Life depends on DNA. Remember these three important components of the DNA molecule. First, DNA stores the information that a cell needs to produce proteins. Second, DNA stores the information required for cells to function. And finally, third, DNA is passed down from generation through generation. As we previously mentioned, the DNA molecule is in the form of a double helix, and this structure was first described officially in 1953. James Watson and Francis Crick are credited with deducing DNA structure, but they didn't do it alone. They relied upon the evidence from many other scientists that had been working on this problem for many years. For example, Erwin Chargaff showed that DNA contains equal amounts of A, which is adenine, and T, which is thymine, as well as equal amounts of G, which is guanine, and C, which is cytosine. Recall that these are the so-called nucleotides, or base pairs, that make up DNA. Second, Maurice Wilkins and Rosalind Franklin used a technique called X-ray diffraction to determine DNA's three-dimensional shape, which is a huge part of that double helix. There's a bit of drama behind this history of the structure of DNA discovery. Back in the 1960s, these guys, Watson, Crick, and Wilkins, all received the Nobel Prize for DNA's structure. However, Rosalind Franklin was really the person that did a lot of the work. However, her boss basically took her work without her permission and gave it to Watson and Crick. She died in 1958 of natural causes, and so she could not be awarded the Nobel Prize because she had died before it was given for the discovery of DNA structure. What is DNA? Well, the technical term is deoxyribonucleic acid, and here we can see that full name on our screen. It's composed of a nucleotide that has a phosphate group, a sugar, and a nitrogenous base. And in the case of DNA, the sugar is called a deoxyribose. DNA is composed of nucleotide monomers, and it follows some base pairing rules. A binds to T, and G binds to C. DNA resembles a twisted ladder, and the rails of the ladder are these sugar-phosphate backbones. Two chains are parallel but oriented in opposite directions, kind of like the northbound and southbound lanes of a highway. Hydrogen bonds connect complementary DNA strands. Special types of covalent bonds connect the sugar backbone to its nitrogenous base. Human cells contain about 6.4 billion base pairs in their nucleus. The human genome is defined as all of the genetic material found in the cells of a living organism. And remember, there's organelles that contain DNA. We've already talked about the nucleus. That's where we find the nuclear DNA. But both the mitochondria and the chloroplast also have their own unique DNA code that is separate from the nuclear DNA. When we're studying for the exam, make certain you can describe the components of DNA and its three-dimensional structure. DNA stores information. A gene is a sequence of DNA that encodes for a specific protein. And we can think about these proteins as really representing traits in organisms. Right now, we want to consider just how it is that we can go from DNA to protein, and this relationship is called the central dogma of biology. Another fun fact, the human genome includes anywhere from 20,000 to 25,000 genes. So the central dogma is this process of going from DNA to RNA to protein. It involves these transformations of information. Stage 1 is transcription, in which the information stored in DNA is copied to RNA. Where does this occur? It happens in the cell's nucleus. Stage 2 is translation. Translation is the information in DNA is finally used to build our protein, and that protein represents a trait in that organism. Where does this occur? It occurs in the cytoplasm of the cell, outside of the nucleus. In this table from your textbook, it shows us some differences between DNA and RNA. They differ in sugar, they differ in nucleotide bases in that DNA has T, or thymine, and RNA has U, or uracil. They differ in form in that DNA is generally double-stranded, but RNA is generally single-stranded. They function differently as well. The function of DNA is to store RNA and protein encoding information. In essence, DNA transfers information to the next generation of cells. RNA carries protein encoding information. It helps the cell to make proteins, and in some cases actually catalyzes reactions. Recall we learned that enzymes catalyze reactions, but in some cases, RNA will catalyze reactions as well. RNA is this very important middle step in the flow of genetic information. It's so important that RNA comes with 3 different types. We have the messenger RNA, the ribosomal RNA, and the transfer RNA. Let's take a look at ribosomal RNA for just a moment. Ribosomal RNA forms part of a ribosome, which is the physical location of protein synthesis. There's a web address here on your screen. Make sure that you play around with this website at home. It's from the Learn Genetics website, and it does a great job of showing us all the parts and functions of a cell. Here's our first challenge question to make sure you understand what we're talking about. The question is, what is the main function of DNA? The first choice is to encode proteins, or to produce ATP, or to speed up cell reactions, or to provide structural support to the cell. And finally, are all of these correct? Pause the lecture if you don't want to hear the answer. The only main function is simply to encode proteins. DNA primarily serves to encode proteins. When you're studying for the exam, make certain you can describe the central dogma of biology by describing the relationship between a gene and a protein. So now we're going to look at the steps of transcription versus translation one by one. Transcription uses DNA to create RNA. Let's look at how the cell produces this RNA copy. Recall that transcription occurs in the nucleus. DNA pairs with RNA according to base pairing rules. A pairs with U, C pairs with G, G pairs with C, and T pairs with A. Now notice there is no T in the RNA. There's no thymine in the RNA. Instead, RNA only has U, or uracil. Transcription occurs in 3 distinct steps. Step 1 is initiation, step 2 is elongation, and step 3 is termination. We will look at these steps now one by one on the following slides. The first part of initiation involves RNA polymerase binding to the promoter, which is the beginning of our gene. Many different enzymes swoop in and unzip the DNA. Please note that in this figure from the textbook, it doesn't show these enzymes, but we can always assume that whenever chemical reactions are occurring in cells, there's a lot of enzymes that are involved. The DNA template strand encodes the RNA molecule according to the base pairing rules. RNA polymerase is the first part of elongation. Now we can actually start to build the RNA copy. RNA polymerase is a type of enzyme that moves along our template strand, making an RNA copy. Notice that the RNA molecule is complementary to the DNA template strand. The final step of transcription is termination, in which RNA polymerase reaches the terminator, which is the end of our gene. RNA, DNA, and the RNA polymerase separate. DNA becomes a double helix again, and now the RNA can go on into the next step of this process. The cell has produced a RNA copy of a gene, which is very important to get us set up for the translation process. Before we can move on to translation, the RNA must be processed inside the nucleus. For example, a poly A tail and a messenger RNA cap are added to the RNA molecule. In addition, we have to do the splicing of our RNA. During splicing, introns are removed from the RNA molecule. The RNA is now ready to leave the nucleus and move onward to translation. When you're studying for the exam, make certain that you understand how messenger RNA is modified before it leaves the nucleus of a eukaryotic cell. Now we can move on to the next phase, which is translation. Translation does the business of building our protein. Now let's look at how the ribosomes play a specific role in using RNA to produce this protein. And recall, all of this translation process occurs in the cytoplasm outside of our cell's nucleus. We need to know this term now, which is a codon. Codons are 3 nucleotide sequences that encode for 1 amino acid. We can see that the DNA was transcribed into a messenger RNA, and we know that messenger RNA is translated into a protein. Well, this translation occurs in the form of this language of codons. That is to say, codons can be attached to a growing polypeptide, which is essentially an amino acid sequence. The genetic code corresponds to messenger RNAs, which correspond to amino acids. So on the table from your textbook, it shows us all of the possible combinations of these amino acids that can give us these codons. Here's a challenge question. If we mutated a gene and we changed the order of these amino acids within a codon, would this mutation always cause changes in the trait for that organism? What do you think? Yes or no? Pause the lecture if you don't want to hear the answer yet. The answer is no, because notice that for a given codon, there are multiple codons that correspond to that amino acid. And so because of this, just changing one letter isn't necessarily going to completely change the amino acid. Translation does this business of building the protein in that transfer RNA molecules translate the genetic code. Transfer RNA binds to a messenger RNA codon, and it occurs at the anticodon end of our transfer RNA molecule. In addition, we have a binding of our amino acid at the other end of that transfer RNA. Translation occurs in 3 steps in the same way that transcription did. It's initiation, elongation, and termination. And all of these steps happen directly in relation to the ribosomes. During initiation, the small ribosomal subunit will bind to the messenger RNA. In addition, a large ribosomal subunit will bind, and the first transfer RNA molecule will bind as well. All of our players swoop in to get us ready for the translation process. Transfer RNA complementary base pairs to the messenger RNA. Please note that the transfer RNA already carries an amino acid, which is called methionine, or MET. This is the start codon that gets the whole process going. The second transfer RNA enters the ribosome next to the first transfer RNA. Amino acids covalently bond, and we're starting to grow our polypeptide chain. The first transfer RNA leaves, and the ribosome moves to the right, and yet another transfer RNA swoops in, carrying the corresponding anticodon to the codon. But notice that the amino acids remain bonded together. This process continues and the protein grows until eventually we have a long, fully formed polypeptide chain. In termination, the ribosome reaches the stop codon. A release factor binds and the polypeptide chain releases. It detaches from the messenger RNA and folds up into a fully functional 3-dimensional protein. Translation is very efficient when multiple ribosomes attach to a messenger RNA molecule simultaneously. When you're studying for the exam, make sure that you understand all the steps of transcription and translation and where in the cell transcription and translation occur. Protein synthesis is highly regulated because it takes a lot of energy. Cells can save energy by only producing proteins when they're needed. It's important to note that both prokaryotes and eukaryotes regulate protein synthesis in very different ways. Now we're going to learn about mutations and how mutations can change the DNA in an organism. For example, a mutation in one gene can cause a fly to develop legs where its antenna should be. A mutation is a change in a cell's DNA sequence, and mutations come in several varieties. They range from silent, meaning they have no effect, to devastating, meaning they most likely kill the organism. A point mutation changes one or a few base pairs in a gene. The table in your textbook does a great job of showing us how we can use sentences to represent codons and use this to show how point mutations can make a difference. Remember that codons are sequences of 3 nucleotides. Each word in these sentences represent one single codon. The wild type is the original nucleotide sequence. Substitutions involve changed nucleotides, either one substitution or sometimes multiple substitutions may occur. Deletion involves nucleotides being deleted from the code. And again, it could be one deletion or several deletions from that code. In addition, we can have insertions in which nucleotides may be added to our code. Frameshift mutations have the most likelihood of causing a big effect on the organism's DNA because frameshift mutations affect multiple codons. Insertion of one nucleotide changes every single codon that comes after that insertion in this chain of letters. Notice in the frameshift sentence, it doesn't even read like a sentence anymore. It looks like nonsense because this frameshift has affected everything past the insertion. Deletions or insertions of 3 nucleotides add or delete entire codons, but they do not affect the other codons around them. While mutations may change DNA, there are some things to keep in mind. For example, number 1, mutations are random and often spontaneous. Number 2, not all mutations matter to natural selection or evolution. And finally, number 3, if environmental conditions induce a mutation, for example, if you get exposed to UV radiation or pollution, that mutation will not necessarily give you a favorable effect. It's also important to note that we can't just mutate on demand to allow us to survive a changing environment. A single base substitution in the human hemoglobin gene causes blood cells to form abnormally, which may lead to sickle cell disease. This is an example of a harmful mutation that has some unexpected benefits. We will explain these benefits once we get into our next part of this lecture series in a later chapter. However, not all mutations are harmful. Mutations create different versions of alleles, and we know that alleles are alternative versions of the same gene. Genetic variation is very important for evolution. It provides the raw material for natural selection to act upon. Plant breeders might even induce mutations to create new varieties of plants, such as bigger, juicier grapefruits, or grasses such as this wheat that are resistant to pests, or cotton plants that produce larger, fluffier cotton so that we can more effectively make our clothing. When you're studying for the exam, make certain you know what the types of mutations are and how these mutations might affect encoded proteins. The last part of this lecture is on viruses. Viruses are not living things. Viruses are simply genes wrapped in protein coats. We know that cells use genes to produce proteins, but if a cell receives new genes, will it produce new proteins? To answer the question, let's take a look at viruses, which inject genes into their host cells. Here we see a huge variety of viruses They come in a huge variety of shapes and sizes. However, it's important to note, viruses cannot reproduce unless they enter a host cell. That is why they are not considered living things. Viral genes contain instructions for making more viruses. So therefore, viral replication involves the following steps. Number 1, the virus attaches to the host. Number 2, the virus penetrates the cell and releases its DNA. Number 3, the host doesn't know any better. It takes the virus DNA and it transcribes it and translates the virus DNA as if it were its own. Number 4, new viruses assemble. And number 5, the viruses leave the cell where they can go out into other cells and infect even more. Here's another challenge question. True or false? Cells will express foreign DNA, such as DNA injected by viruses. Pause the lecture if you don't want to hear the answer yet. It's true. Viruses can also cause diseases in plants. The color patterns you see on your screen here, the leaf has a bunch of spots on it and the flower has a bunch of streaks in it. They might look pretty, but they're actually caused by viruses in this plant. Drugs and vaccines can help us fight viral infections. Vaccines teach your immune system to recognize a virus. Antiviral drugs, however, are very difficult to develop because viruses have a very high mutation rate. It's also important to note that antibiotics do not work against viruses. They only work against bacteria. In the last part of our textbook chapter, it shows us this investigating life question about clues to the origin of language. It shows us the rhesus monkey, the gorilla, the chimpanzee, and the humans. And we can see that few amino acids differ between the language gene found of humans, primates, and mice. 2 recent mutations in the human genome might set our language skills apart from other primates. As a final post-assessment question, you should be absolutely certain you understand this question. Which of the following statements is true about the occurrence of mutations in a population of organisms? The first choice is mutations that give favorable effects will occur more than mutations giving unfavorable effects. Your next choice is mutations that give favorable effects are more likely to remain in a population over time than mutations giving unfavorable effects. The next choice is natural selection increases the overall rate of mutations. And your last choice is unfavorable environmental conditions increase the probability that new mutations will give a favorable effect. So I'd like you to really think about this for a moment. Please pause the lecture before you see the answer revealed. The answer is B. The only true statement in this list is that mutations that give favorable effects are more likely to remain in a population over time than mutations giving unfavorable effects. It's very important that you note that the last choice is false. Unfavorable or bad environmental conditions do not increase the probability that new mutations will give you some selective advantage. For organisms to survive a changing environment, they need to already have those mutations in their genome and already have those traits